Hello dear friends, welcome to my channel Mithul Bioblog. How are you all? Hope you all are safe. Today I am with a video. It's a continuation of my previous video about the viruses. Today I will be explaining about the classification of viruses based upon the criteria type of host. Actually, we can classify viruses based upon many criteria like mode of that is type of nucleic acid, cause of infection, shape of viruses, and so on. Today we can see how viruses are classified based upon the type of host. Based upon this criteria, viruses are classified into three types. One is plant viruses, second is animal viruses, and the third one is bacterial viruses. Otherwise, it is called as bacteriophage. Let us have a comparative study on this. All these three viruses are pathogenic. It means they are disease-causing form. If you tell about the shape of the viruses, plant viruses are rod-shaped, whereas animal viruses are polyhedral or spherical in shape. Bacteriophages are tadpole-like structure. Tadpole we know, it is the baby form or nymph form of frog. Now if we tell about the genome, plant viruses contain RNA, whereas animal viruses, they are containing double-stranded DNA or single-stranded RNA or even double-stranded RNA forms. Bacteriophage genome, it's of DNA and RNA type. Usually we can see double-stranded DNA forms. Now if we tell about the diseases caused by this type of viruses, few examples I have mentioned here. At least we should know that, especially this +1, +2 batch students and even high school section students. Plant viruses causes diseases like tobacco mosaic disease, tomato leaf curl, bunchy top of banana. Animal viruses cause diseases like foot and mouth disease of cattle, rabies, AIDS in human, Ebola, COVID-19, and also there are many diseases which you have to study in the 9th standard syllabus and even in +1 like polio, chickenpox, smallpox, influenza, measles, etc. Now next is about the bacteriophage. Actually, if we are asking whether they cause diseases, they are not showing any direct effect on humans in causing disease, but indirectly they are the reason for many diseases like cholera, diphtheria, etc. As we all know, these bacteriophages are virus which infect these bacteria. So this happens so. Okay children, now let us study the structure of bacteriophage. Very important question used to come for exam, especially in the high school section and +1 syllabus. Okay, the structure of bacteriophage can be divided into three main parts. The first one is head. Head region contains the capsid and the core. Capsid is a protein core and core is a nucleic acid which is seen inside the capsid. Second portion is the neck. Neck is called as collar. Then comes the tail. Tail is surrounded by a contractile sheath. Towards the base of it, there is a base plate with tail fibers. And this part is called as host recognition sensors. Why? Because they recognize the specific host and cause infection. So this is the structure of bacteriophage. You should know to draw the structure of bacteriophage and also mark the part. So in short, bacteriophage is containing a head region which is made up of capsid and core. Then comes the neck which is called as collar. Then tail and base plate with tail fiber. Okay children, next let us study a special type of virus called as retroviruses. Retroviruses are the RNA virus and their action is entirely different from that of the DNA forms. So before telling that, let me tell how the DNA acts in the body. The action of DNA. DNA, as we all know that DNA is a genetic material in our body and they carry the genetic information. DNA can undergo a process called replication, that is DNA synthesis. DNA can synthesize themselves. After that, the DNA undergoes the second process that is called as transcription. Transcription is a process in which DNA synthesizes RNA. So that is the second step, DNA synthesizes RNA. After the formation of RNA undergoes the third process, which is called as translation process. Translation is a process in which RNA synthesizes protein. And in this way, genetic information is transferred from DNA to RNA and from the RNA to the protein. And protein play a very important role in our body because they carry out the function of our body. So that is the relationship between all these three things. Here we can say RNA, in the case of viral RNA, that is the RNA viruses, the action is entirely different. Now how they act? This RNA viruses, which contain the viral RNA genome, they undergo a reverse transcription process by which they synthesizes a viral DNA. So this is an opposite action of transcription. Once this viral DNA is formed from viral RNA, they enter into the host nucleus and they undergo the further process and numerous retroviruses are formed. So this is the action of retroviruses inside a body or inside a host cell. Okay children, now let us study the life cycle of virus. Are you ready? Life cycle of virus is completed in 5 different steps. They include attachment, penetration, biosynthesis, maturation and lysis. Now let us study this in one by one. First of all, attachment. The virus come and attach to the host body. That is the first step. Then next is penetration. In the case of DNA viruses, once they attach the host body, they inject the viral DNA into the host and the capsid is left outside. It means the complete virus will not enter into the host body. Only the viral DNA is injected into the host body. Whereas in the case of RNA viruses, which is having envelope, the capsid with envelope enter into the host body by a process called endocytosis, and then only the further changes take place from inside the host. Clear? The next step is biosynthesis. Here, the viral genome, once entered into the host, they undergo the different process like replication, transcription and translation. After that, finally a specific protein will be formed. Already I have explained replication, transcription and translation process. So this is happening in biosynthesis process. Once the genome is entered, the protein is formed finally. Okay. Now the third step is maturation. Once the protein is formed, by using this protein, capsid is formed for the formation of virus. Capsid, once it is formed, they integrate with the viral genome and as a result, new viral particles are formed. The last step is lysis. Once the new viral particles are formed, they are released from the cell or released from the host. So these are the steps in life cycle of virus. I hope it is clear to you. So that is all for today. I hope everything is understood by you. If you're having any doubt, you can ask me in the comment box. Okay children, so that is all for today. Thank you for watching my video. And if you like my video, please subscribe, share and like. And only one thing to say, keep calm, stay home, be safe. Okay, bye-bye. See you. Take care.